Hi everyone, this is Dr. Jenkins, and I wanted to put together a little bit of a review since we were so rushed in lab this week. I want to put together a little review. This video hopefully won't be too long, but I want to make sure that you know exactly what to review and what to study. I'm going to make this PowerPoint available to you. I've also put together a Word document that just outlines exactly what you need to review for our first lab quiz. If you remember, lab number 1 was our lab on organization of the body, and then we did the rat dissection. So I've taken this right from the Word document that I'm going to make available to you. So make sure that you review all the things that we reviewed in lab. So these are all the things that we reviewed in lab, right? Notice I made some little hints for you. Be able to compare body regions using these directional terms. We practice that in lab. Be able to do that for the upcoming lab quiz. With the movement terms, flexion, extension, abduction, adduction, be able to look at a picture and describe what motion is occurring at each joint. We did that too. We practiced in lab. Okay? Lastly, down here, be able to label the organs on a color picture of the rat. There are review pictures on Brightspace. I can't guarantee it'll be the same picture, but I'm going to provide for you a color picture of the rat, similar, if not the same, as what we see in Brightspace. What I want to spend most of the time in this brief video is on lab number 2, which we really had to rush through. Basically, this is exactly what I put on the board in lab before we were rushing to leave. But here what I've done is I've added the definition. Now, you're going to go through this with lecture with Dr. Mastrangelo, but I want to make sure that you hear me talk about it in the words that I use. Sometimes different instructors tend to use slightly different ways of saying things. So just for continuity's sake, I think it's beneficial for you to hear me review this so you can prepare for the lab quiz that I'm making, right? So I'm going to be pretty quick about this because you're also going to get this in lecture. Make sure you review the phospholipid bilayer and selective permeability. The phospholipid bilayer, this should sound familiar and look familiar. You're going to talk about the cell with Dr. Mastrangelo in lecture. And if we zoom in on the cell membrane, let me make this wider. If we zoom in on just that outer covering, it actually, in reality, is a phospholipid bilayer. Each little thing like this is a phospholipid. They kind of look like the pegs from the board game Life. But they're arranged in a bilayer where there's a double layer, right? And you'll know from lecture that the heads of each phospholipid face the inside and outside of the cell, and the tails face the middle. Okay. The reason why we have this seemingly complex outer cell membrane is that it is selectively permeable, which means that, in this case, the phospholipid bilayer is a membrane that is semi-permeable. In other words, it allows some things to pass through it, but not all. So it is selective about its permeability. Of course, we're going to need to bring some things into the cell. Don't we need to bring in oxygen, glucose, etc.? Likewise, don't we need to be able to remove some things out of the cell? Waste products, CO2. So in other words, we have to have this membrane that allows us to move some things through, but yet at the same time, we don't need it to be just open to everything, because then we would be letting in bacteria and other things. Good. Then make sure you review some ways in which we can move things through this plasma membrane, the phospholipid bilayer. This is all a review, folks. Diffusion, osmosis, active transport. You know this. Diffusion. Movement of molecules or particles from an area of high concentration to low concentration. What if we have 50 particles on one side of the phospholipid bilayer and we have 10 here? There's a gradient. On one side of the membrane, there's a different amount than the other side. Diffusion means that things move from high to low. High to low. Make sure you review, there is simple diffusion. Simple diffusion is when things move directly through the phospholipids, and that's what this picture is showing you. Examples of things that move through simple diffusion would be gases, like oxygen, carbon dioxide. But sometimes a particle is too big, and it needs to move through facilitated diffusion. Facilitated diffusion is still moving particles from areas of high to low concentration, but it's just doing so through a protein channel. So the diffusion is facilitated or helped by a channel. Larger particles tend to do that. The examples that I'm giving you are ions and glucose. It's still diffusion from high to low, but they diffuse through a protein channel, which is specific, right? Glucose can only diffuse through a glucose-specific protein channel. Sodium can only diffuse through a sodium-specific protein channel. I want to make sure that we point out, you already know this, I'm sure, these are all passive. Diffusion is passive. It does not require energy. If there's a gradient, and a selectively permeable membrane in between. If there's a gradient and a selectively or semi-permeable membrane in between, things will move from high to low. Diffusion is passive. Osmosis, simply put, is diffusion of water. Movement of water from high to low. Movement of water, it's the same principle. There's a gradient. There's more particles on one side than the other. Right? In this picture, it's not the best picture. In this picture, there's 5 water molecules on this side and only 4 on this side. It's not a big gradient, but it is different. If there's a difference between the two sides, and if there's a semi-permeable or selectively permeable membrane in between, things will move from high to low. When we talk about water, we tend to use the word osmosis, still passive. Sometimes water will diffuse simply right through the phospholipids. Sometimes water will move through a channel. Good. Then we get to active transport. Diffusion, whether it's simple diffusion or facilitated diffusion, this is another picture showing the same thing. Whether it's simple, this is a picture of simple diffusion. Whether it's simple diffusion or facilitated diffusion, it's passive. Passive goes from high concentration to low concentration. That's why it's passive. It's like you're going downstream, down the current. Easy. However, sometimes we need to move things from low to high. When we move things from low to high concentration, it's called active transport. Why is it called active transport? Because it requires energy. And what's our main energy source on the cellular level? ATP. If I'm going from 2 particles here, I'm not going to count these, but let's just say it's 10, a little more than 10. If I'm going from low to high, I'm going to need energy. I'm going against the gradient. It's like paddling upstream, up current. But sometimes, as is the case in the sodium-potassium pump, sometimes we need to do that. Good. So I have covered or reviewed, all those things. Let's move on to the last part here, Brownian motion, osmotic pressure, and tonicity. Make sure you review Brownian motion. You can just go to YouTube, type it in, you can see it. Brownian motion is a random vibratory motion. Particles, by nature, have a random. There's plenty of ways. Vibratory. vibratory motion. There's plenty of ways to define this. Use your own term. For some reason, vibratory seems to click with me. Particles are just jumpy. Particles are jumpy. This tends to help diffusion. Diffusion is still passive, folks. Diffusion is still passive, but the fact that these particles tend to have a natural vibratory energy to them tends to help diffusion. Make sure you review osmotic pressure. You can see I've got more words on these slides because I pulled this from one of my lecture presentations. But I've put in red and circled the most important things. Osmotic pressure is a type of fluid pressure that can build up on one side of a membrane. So it's a fluid pressure that's caused by a buildup of solutes on one side of a semipermeable membrane. And we care about this because it's a force. It's a pressure. It's a force against the cell membrane, against that phospholipid bilayer, and it can pull fluid in one direction. So it's kind of hard to see because it didn't come over very well, but we tend to move water in and out of a cell membrane in two ways: osmosis from high to low, but also through this osmotic pressure. We're going to come back to this later in the semester, particularly with Dr. Mastrangelo in AMP 2, when we get to blood vessels and capillary exchange and all this other stuff. We're going to be talking a lot, the kidneys, oh my God, the kidneys. We're going to be talking a lot about this osmotic pressure. For now, let's just simply define it, put it in your memory bank, and that way when we come back to it later, it will sound familiar. So osmotic pressure, it's a fluid pressure that can build up on one side of a membrane. And it means that we can pull fluid in one direction. I'm just giving you these bullet points, folks. We're going to be utilizing this principle of osmotic pressure in discussing tonicity. You're going to get this with Dr. Mastrangelo too. Now, there's so many ways to define tonicity, but I like to define it this way, what I have circled it in red, because I think this is a good starting point. Tonicity, it's really a characteristic, how the shape of a cell may change. Our examples of tonicity are going to be, sometimes a cell can get bigger if it takes on a lot of water. So another way of saying that is, sometimes if a lot of water diffuses into a cell, wouldn't that cell get bigger? It's really nothing earth-shattering. If all of a sudden a lot of water diffuses into a cell, that cell's going to probably get bigger. The opposite can also happen. What if a lot of water diffuses out of the cell? If a lot of water leaves the cell, that cell might get smaller. All related to this is this principle called tonicity, how a cell may change shape, largely based off of what's diffusing into and out of it, but also based on, ding, 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 this osmotic pressure. What we're going to find, again, you need to focus on what I have circled. What we're going to find is water moves towards a higher solute area. Okay, I'm giving you the bullet points that are most important. Water moves towards high solute. So if we were in the lab, we would have done this experiment where we put red blood cells in water. It could have been in regular water, it could have been in a saline solution, and it could have been in a higher salt solution like seawater. In some instances, if you put red blood cells into a beaker of, this is actually saline, so it's 99.1% water and just a touch of salt, 0.9% salt. In this case, the cell will stay the same shape. So among tonicity, there's three possibilities. If a red blood cell maintains the same shape, that solution is said to be isotonic. Look, these red blood cells look normal. If a red blood cell stays the same shape, the solution is said to be isotonic. If the cell gets bigger, If the cell gets bigger, look what's happening here, folks. Cells are getting bigger. That solution is said to be hypotonic. If a cell gets smaller, look here, the cells are kind of crinkling. You can see that outer edge is ragged. If a cell gets smaller, it's said to be a hypertonic solution. Just be able to define those. I could ask you a simple question. What kind of solution is it if a red blood cell maintains the same shape? Isotonic. I could ask you, what kind of solution is it if a cell gets smaller? Hypertonic. It can be a little counterintuitive because doesn't the prefix hyper mean more? That's true. It's named based off of the solute, which we're going to talk about in a minute. Some people like to go through the entire thought process to remember that. To start this off, I just memorize it as hyper, the cell gets smaller. The way that I remember that is if someone is hyper, they may go see a shrink. Haha, if you're hyper, you may go see a shrink. Now, in lab, we would have talked about this more, and you're going to get this in lecture too. Here is a case where we've put a red blood cell into a salty solution. So this fluid is 3% salt. Seawater can be up to 3.5%. So imagine you're in the ocean, right? Well, based off of that osmotic pressure and regular diffusion, that cell will get smaller. So what are the things to remember? In a hypertonic solution, the cell gets smaller. By the way, the fancy word for get smaller is called crenates. For lab, you don't need to worry about where there's more solute, where there's not. You might for lecture, but I'm only talking about for lab. What are the things you need to know for lab? Be able to define tonicity, how a cell may change shape. Okay, so things to review for your lab quiz. Number 1, define tonicity. For us, we're going to define it as how a cell may change shape. Number 2, be able to identify in what kind of solution does a cell get bigger, smaller, or stay the same size. In a hypertonic solution, cell gets smaller. In a hypotonic solution, cell gets bigger. And in an isotonic solution, cell stays the same size. What I want to point out here is, remember I said water moves towards or is pulled towards higher solute. I'm going to go back, and I hope I don't give you vertigo with going back and forth. Water moves towards higher solutes. This is what I'm talking about with that pulling. Water moves towards higher solute. So that's the third thing to remember. Water moves towards a higher solute. So in this example, where is there a higher solute? The solute is what's being dissolved, or it's what's present in smaller amounts. What's the solute here? Sodium chloride. Is there more sodium inside the cell, or is there more sodium outside the cell? We have 0.9% sodium inside the red blood cell, 0.9%, and 3% sodium outside. There's more solute outside the cell. Water moves towards higher solute. So how is that water going to move? Water's going to move towards where there's more solute. If all this water leaves the cell, what's going to happen to it? It shrinks. Why does the cell get bigger here? Because water is moving towards the area of higher solute. Water is moving towards the area of higher solute. Where is there more solute? Inside the cell. Here, there's no solute, 0. So even though it's not much of a difference, compared to these two places in the beaker and inside the cell, there's more solute inside the cell. Water's going to move towards area of higher solute. If water comes into the cell gets bigger. By the way, the cell may take on so much water, it may burst. The fancy word for bursting is hemolyze or hemolysis. Obviously, as a side note, this is bad. We don't want our red blood cells to take on water and get bigger and burst, because then they can't do their job. This is why we want, if we're giving someone a fluid IV for hydration, we want it to be saline, because saline is isotonic to the blood. So we can hydrate the bloodstream without causing the red blood cell to get smaller or get bigger. Okay, I'm going to quickly go through these last parts. The last thing to review for your lab quiz would be what we would have done in lab. And I mentioned this on the board. I told you we were looking at the influence of 3 things on diffusion. Now that we have defined diffusion, now let's talk about what influences diffusion, the rate and the amount. Concentration gradients, one thing we looked at, particle size, and particle weight. Make sure that you know the relationship between these. Number 1 is concentration gradient. We already talked about it, so there's no sense in hiding the fact. When there's a bigger concentration gradient, the larger the concentration gradient, the more diffusion. If there's a lower concentration gradient, there's less diffusion. This here is outlining what we would have done in the lab, if you're interested. We would have had a beaker of water. Go for blue for water. We would have had a beaker of water, and into each beaker of water, we put a little dialysis tubing bag of Caro. Caro is another word for sugar. So we had this little dialysis tubing bag. That's what this is. Here's a picture of dialysis tubing. It kind of looks like it's clear, but up close, microscopically, there's tiny pores. So it is a semi-permeable membrane. By the way, it is called dialysis tubing because it is what they use in a dialysis machine. And because a dialysis machine acts like a kidney, we need that membrane to be semi-permeable so we can move things into and out of that tubing bag. In the lab, we would have made these 3 bags of sugar, and these sugar bags were a different concentration of sugar. We had a 25% sugar, meaning that bag had a fluid that was 25% sugar and 75% water. A 50% sugar bag, meaning inside the bag was 50% sugar, 50% water, and a 75% bag, which had 75% Caro, 25% sugar. I would have asked you, will the bags gain or lose weight, and which will gain the most weight? If we think about this, let's think about the concentration gradient. So let's look at the first one. Inside the bag is 25% Caro. If the bag is 25% Caro, the rest of the fluid in that bag is 75% water. The outside, the beaker, is all water. So here we have a gradient of 75% water to 100% water. We're looking at the gradient. Here it is 100 to 75. What's it here? If this Caro bag is 50% sugar, it's 50% water to the same 100% water. So what's our gradient here? 100 to 50. Here, the last one, it's 75% Caro, which means inside that bag is 25% water to 100% water. So what's the gradient? 100 to 25. I hope I'm doing myself justice here. It's always better when we're in person, but here are our gradients. 100 to 75, 100 to 50, and 125. Which has the biggest gradient? Well, obviously, the biggest gradient is this one. So we would expect this bag to gain the most weight. Water's going to move from high to low. Water's going to move from high to low, 100% water to less. And so each bag's going to gain weight because there's more water outside the bag than inside. Which is going to gain the most weight? It should be this one because it has the biggest gradient. Was that true? Yes. You don't have to know about these gradients and all that crap. But if we look at the total weight gain, I have the numbers because I did the math. This had a total weight gain of 0.1 grams. This one, 0.18 grams. This one, 0.23 grams. Which had the biggest gain in weight? This one. I would like you to know that. Hope you've been paying attention. Which bag would have gained the most weight? 75% sugar. We were looking at that bag. It had 20. This one had 25. Let me say that over again. This particular bag had 75% sugar, or caro, which means it had only 25% water. This bag gained the most weight because it had the least amount of water. So there was the biggest concentration gradient. Okay, then we would have looked at particle size. Here, we would have made a beaker of water and also had a dialysis tubing bag, but this time we would have filled the bag with a solution containing 3 things: sugar, salt, and starch. In this one, we were looking to see, of these 3, sugar, salt, and starch, which would be able to diffuse out of the bag. This is a matter of size. Sugar and salt are a little bit smaller. Starch is big. What we would have found is that sugar and salt were able to diffuse out of the bag, but starch was not able to diffuse out of the bag. Why? It's too big. So even though that dialysis tubing bag is semi-permeable, this is too big. It just couldn't fit. So this is what I said before, smaller particles tend to diffuse more because they're more likely to fit through the semi-permeable membrane. I would like you to know, of these 3, sugar, salt, and starch, which ones were able to diffuse out of the bag? Sugar and salt. Which one was not able to diffuse out of the bag? Starch. Why? Too big. And lastly, we would have looked at particle weight, not always associated with size. We already know the relationship because we talked about it. Particles that are lighter tend to diffuse more. Why? Because they have more of that Brownian motion. Lighter particles tend to have more diffusion. We would have seen it before our very eyes. We would have taken an agar plate and put 3 different color dyes: the methylene blue, the malachite green, and the potassium permanganate. They have different weights. Heaviest is the methylene blue, middle, malachite green, and the lightest is the potassium permanganate. Over time, which do we expect to diffuse the most? The potassium. Why? Because it's lighter. And sure enough, this is a really bad demonstration. This one diffused the most. It got bigger. Lighter particles diffuse more. Perfect. All right, folks, that's the review. Any questions, email me, and I'll see you next week.